So this week is our cell membrane week. So there's a lot going on here because the cell membranes are really important for us to understand if we want to understand how cells connect to each other, how materials move in and out of cells, and the forces that drive that process of moving stuff in and out of the cell, how cells protect themselves, right? This is some really important stuff for understanding how cells function, is understanding how the cell membrane works, okay? Both its structure, which gives us information about its function, right? Structure and function, form and function are tightly linked in living organisms. Okay, so let's dive in. First, we're going to talk about the membrane structure. This will be a little bit of review from last week when we talked about cells, but we're going to dive in even deeper into the cell membrane. Okay, so this is an image that we've seen before. Okay, so just want to reorient ourselves. That cell membrane is that layer that surrounds everything inside the cell from the outside world. Okay, so we typically look at just the phospholipid bilayer, but it is really complex. There's a lot going on here. We have collagen fibers and polysaccharides going on out here, this extracellular matrix that helps the cell stick to other cells. It helps with signaling, it helps with coordinating cellular activities. Then in our phospholipid bilayer, which actually makes up the membrane itself, right? Phospholipid bilayer, we have proteins that are embedded in it. These proteins have a wide variety of functions, which we'll talk about in just a minute. Some of these proteins are anchored to microfilaments of the cytoskeleton. Remember the skeleton that gives a cell its structure and helps it move, right? Some of these are anchored to that cytoskeleton. We see also some glycolipids or some glycoproteins. So these are carbohydrates and lipids connected together, or carbohydrates and proteins connected together. Those are really important for cell-to-cell communication, cell recognition, things like that. All right, so let's jump in. But just keep in mind that this side down here, the cytoplasmic side, that's the side that's inside the cell. Okay, so that's where the cytoplasm is. And this side out here, the extracellular side, extracellular just means outside the cell. Okay. So the main component of that phospholipid bilayer, of that fluid mosaic like we called it, right, is the phospholipid molecule, right? The main component of the membrane is that phospholipid molecule. We've talked about this molecule a lot, and there's a reason behind that. So phospholipids are what we call amphipathic, meaning that they kind of had two lives. They have their hydrophobic life, which is in these fatty acid tails right here, right? Those carbon-hydrogen chains, those are very nonpolar, those are very hydrophobic. Then they have their hydrophilic life, right? So they have their phosphate head up here, which is very polar, okay? So it's a very negatively charged functional group, so it's very polar. So it likes to interact with water, it's very hydrophilic. So when we put phospholipid molecules in water together, they will already, due to that nature of the fatty acid tails being hydrophobic and their phospholipid heads, they will create this bilayer, okay? But another key component of this phospholipids is these tails. What's unique about them? What do we see here, this bend right here? That is because they are unsaturated fats. For at least one of their tail are unsaturated. And remember, unsaturated fats tend to be liquid. They stay liquid, especially at room temperature. Okay, so that's important. This bend in this tail right here is caused by a double bond between some of the carbons. Because this is an unsaturated fatty tail, which helps keep the membrane fluid in a variety of environmental conditions. So membranes are fluid. We show them as kind of static things because that's how we when we're showing images, we can only draw them in one state, but they're fluid, they're moving, okay? They're kind of like a liquid, but they're very viscous, they're a viscous liquid. And the reason why that is because of that double bond in those fatty acid tails. That helps keep them from tightly packing together. Okay, so see how in these phospholipids, they don't have any double bonds in their fatty acid tails. They're totally saturated hydrocarbon tails. that lets them pack together really, really tightly, which would become very viscous or maybe even solid, right? So in saturated fats, the reason why they can become solid is because they don't have any double bonds in their tails, which means they can pack together and form a solid. The double bonds in the unsaturated fatty acid tails keeps these molecules from tightly coalescing and compacting together, which keeps them in a fluid or a liquid form. Okay, so that's the number one thing. It's their fluid and their liquid because they have that double bond in their fatty acid tails, because they have that unsaturated fatty acid tail. And important, we don't want this membrane to be solid. If it's solid, molecules can't move in and out, and that makes it challenging to utilize the membrane. In addition to that unsaturated hydrocarbon, we also have some cholesterol molecules. Remember we talked about cholesterol, it's a steroid. It's a lipid because it's hydrophobic, right? All lipids are hydrophobic, but it has a different shape. It has that four-ring structure, that hydrocarbon four-ring structure. So we see some cholesterol molecules embedded in the membrane as well. These cholesterol molecules help to basically maintain fluidity. They help at low temperatures from the membrane packing together too tightly and basically becoming a solid. And at higher temperatures, it keeps the membrane stable. Okay, it keeps it kind of all connected together at higher temperatures and keeps it from dissociating. Okay, so the cholesterol in the phospholipid bilayer helps to maintain the integrity and the fluidity of that membrane at different temperatures, which is important because our bodies are often at different temperatures, right? Especially if you're an organism that doesn't maintain a constant body temperature, it's really important that your membrane can still function even if you are like just a few degrees above freezing or if you're over 100 degrees. Okay, so embedded in that phospholipid bilayer membrane, we have, remember we call that fluid mosaic because it's fluid, it's liquid, and a lot of times it's moving around. Sometimes the proteins in that fluid mosaic will move around, right? And that mosaic part, the parts that we embed in, right? Think of like a mosaic, like a collage. We take small pieces of other things and we kind of piece them together. That collage part of the fluid mosaic model, or the mosaic part, I guess you could say, are proteins. Proteins that are embedded in that membrane. Okay, so we kind of hit on this a little bit earlier, but what would be the chemical characteristics of a protein that is embedded in that phospholipid bilayer? Remember, these are fatty acid tails, very nonpolar, very hydrophobic. This would be facing outside the cell in the extracellular world, which is usually water. This is facing inside the cell in the cytoplasmic side, which is usually mostly water as well, right? So what types of amino acids would you find here on the cytoplasmic side and here on the extracellular side? You probably would find hydrophilic amino acids, right? Amino acids that can interact readily with water. What about here in this middle portion, where they're interacting with its fatty acid tails? What types of amino acids would you find in this protein? Probably hydrophobic, right? Because they would be, if they were hydrophilic amino acids here, they would not be able to interact and remain embedded in this phospholipid bilayer. Okay, so we tend to find in the middle of the proteins, they tend to be hydrophobic. Okay, in the region that is embedded within that phospholipid bilayer. So there's many different types of proteins all around the cell, hundreds of thousands of proteins in that cell, and different regions on that cell membrane might have different proteins in that area, right? So if you think about a neuron, the part of the neuron that connects to the other neuron has a totally, at the synapse, has a totally different set of proteins embedded in its membrane to help facilitate that communication between the neurons than the parts of the protein that make up the axon, right? So different cells, depending on who they're connected to, what they're doing, what their job is, what tissue they're part of, are going to have different types of proteins in different regions of their membrane. And this can change depending on what's going on in the cell's life, depending on what's going on in the multicellular organism. The types of proteins embedded in that surface are going to change throughout time. Let's just take a overview of some of those proteins and what do they do. So most of these, or a lot of these proteins, are used for transporting molecules across the membrane. So we'll talk about what can move across the membrane and how it moves across the membrane later, but a large portion of these proteins are used to move molecules across the membrane. Some require energy in the form of ATP, adenosine triphosphate, some do not. Okay, but they're there to move stuff across the membrane. Okay, they're kind of like shuttles. They're making sure that there's a place for molecules to move across the membrane, so we call them transport proteins. Some of these proteins are enzymes, all right? So they will, in actual enzymes that grab onto their substrate, react, change the form of that molecule, maybe build it up, break it down, add a phosphate group, do something to it, right? And they will react with substrates either on the outside of the cell, in the extracellular world, or in the cytoplasmic side. So here we're showing the active site here. It's going to react with the substrate, change it from kind of that bow tie shape to the square. That square then activates the next enzyme and it gets changed into a circle. We're showing the active sites right now on the cytoplasmic side, but they very well could be on the extracellular side out here as well. So an example of an enzyme that has its active site on the extracellular side facing out is one of the enzymes that's actually used by the SARS-CoV-2 virus to import into the cell. So here we have the SARS-CoV-2 virus. Okay, we've probably seen some diagrams of this many times before. This red protein structure right here is the spike protein, right? So viruses are basically just a protein shell with some sort of genetic information in them, either RNA or DNA. So what happens, in order for that virus to replicate and make copies, it has to get inside of the host cell. It has to import itself into the host cell, and then it uses the host cell's machinery to copy itself. On some of our cells, we have a large portion, they're very densely populated with this ACE2 protein and this TMPRSS2 enzyme, okay? So these are both enzymes, but they have other functions. So these two enzymes on the surface of the cell have other functions, and they help with vasodilation and vasoconstriction is their main job, usually. But what happens is, and how SARS-CoV-2 has evolved, is that it will bond to the ACE2 protein. And when it does that, this TMPRSS2 enzyme is also right next door. So transmembrane protease serine 2 is what that stands for. So a protease is something that cleaves proteins. It breaks apart proteins, right? So basically what happens is this spike protein will bond to the ACE2, and that then basically activates this transmembrane protein protease serine 2 to cleave off part of that spike protein. And as soon as that happens, as soon as transmembrane protease serine 2 cleaves off part of that spike protein, that basically activates that protein, this spike protein, to import into the cell. So as soon as this enzyme right here cleaves off part of that spike protein, now that virus can import into the cell. So it's using our own enzymes on the surface of our cell to get into our cell, to then hijack our cell's machinery. Okay. So, but that's not what these enzymes have evolved for. They have their own functions. And again, these are involved in vasodilation. One of the vasodilation pathways or vasoconstriction pathways is what they're typically used for. But I just wanted to use this as an example of some proteins or some enzymes on our cell surface that are facing out, that react with their substrate. In this case, the substrate just happens to be, or one of the substrates that they can work on just happens to be the viral coating, the proteins on the viral coat. Okay, so other membrane proteins are going to be signal transduction proteins. So basically what's happening here is that some sort of signaling molecule, could be a hormone, it could be a protein, some sort of communication molecule. It can be picked up by a receptor on the surface of a cell surface. And then that is going to cause a chain of events that will end up by changing either the cell's metabolic processes, changing what's going on inside of the cells, activating or deactivating different enzymes, or even turning on certain genes, activating certain molecules that then turn on genes to be used to build a protein. So the idea is kind of like a game of telephone. One cell sends out this signal, this message, and then that's relayed to the next protein, and then to the next protein, and so on and so forth. So here's an example of a growth factor and how this works. So let's say epidermal growth factor is triggered. We cut ourselves, and our epidermal skin cells need to reproduce. They need to go through mitosis to heal up that cut, right? Epidermal growth factor will be secreted by the cells that are surrounding that cut. That'll be picked up by other cells nearby, by one of these signal transduction proteins, okay? That then causes the phosphorylation of that protein or that enzyme on that cell surface. Often the signal is moved from one molecule to another by adding on a phosphate group. That's how cells kind of talk to each other. They add on or take off phosphate groups. So as soon as that growth factor is picked up by the EGF receptor on the cell surface, it will then activate MKKK, MAP kinase. Kinase is the name of an enzyme. Basically what that does is that then activates another MAP kinase and then another MAP kinase, right? And they activate each other by adding on a phosphate group, just that one phosphate group. And then once that's phosphorylated, it'll phosphorylate another enzyme. And eventually, what happens in this example, it ends up activating what we call a transcription factor. We don't need to know that for right now, but that basically turns on a gene, and it turns on the gene that promotes cell growth and division. So it increases the rate at which cells grow and divide, which means that now we can start to heal that cut that happened, right? Sometimes these signal transduction pathways might end up in changing an ion concentration. So like when muscles contract, they get a signal from a neurotransmitter, which then causes changes in calcium concentration inside the cell in different regions. So they can act in different ways, but usually it's either through phosphorylation or through changing the concentration of ions in different parts of that cell. I'm not expecting you to know the details of this, but the idea of how an external factor, an external molecule can be picked up, and then that transmits the information through a game of telephone, telling this enzyme, then telling that enzyme, then telling that enzyme, which ends up in changing the cell's function in some way. Okay, another type of cell membrane protein. We have glycoproteins, which are a combination of protein with some carbohydrates sticking out, and these are usually used for recognition, from cell to cell recognition. This is how our immune system tells that our cells are ours versus an invader or a pathogen cell, right? So glycoproteins, again, carbohydrate and protein, these are what gives us different blood types, right? Like type A, type B, type AB, type O. We have on the surface of our red blood cells glycoproteins that are embedded that have slightly different shapes to that, especially to that carbohydrate portion of it, right? So this is the protein portion that's embedded in the glycoprotein or the glycolipid. And then based on the structure of that carbohydrate that's coming off, we have type O right here, type A protein, type A, what we call antigen right here. And then here we have type B antigen, okay? And there's some really slight subtle differences, but that's enough for our cells to be able to tell the difference between type A and type B and type O. Okay, so markers on the surface of a cell, we call them, another name that we give them often is antigens, and you'll hear that when you get to talk about the immune system. So glycoproteins are most often used for cell-to-cell recognition of what type cell it is, and is it my cell or is it somebody else's cell? Sometimes these proteins embedded in the membrane are used to hold cells together really, really, really tightly. We'll talk about membrane specializations later, but these long-lasting junctions that keep the cell membrane together are helpful because when we want a watertight barrier, like on our skin, we don't want water getting out or in, right? unless it's coming out through our pores in which we're sweating, these really tight junctions like this help maintain that watertight barrier. Okay, so that's one of the things that they do. So the proteins can grab onto each other and keep the cells really tightly locked together. Other proteins are attached to the extracellular matrix and kind of create a bridge between the extracellular matrix and the cytoskeleton proteins below it, right? And what's really interesting about this is any changes to this extracellular matrix, so if anything's changing in the external environment, or if anything's changing the structure of these proteins, or maybe some of the carbohydrates and the fibers that are coming off of them, that can be transmitted via the cytoskeleton to the rest of the cell, and it can change the cell's processes. Okay, so they're kind of, in a way, like signal transduction proteins, but that's not their main job. Their main job is basically to anchor the extracellular matrix with the cytoskeleton to maintain the cell shape, and it helps keep certain proteins in a specific area as well, right? So we don't want necessarily all the proteins floating around all the time, so these help keep stuff in a very specific location where they're supposed to be. So how do these membranes get embedded in the protein? How do these proteins get embedded in that membrane? How does that happen, right? Well, we kind of talked about this when we talked about how the intermembrane system works. So message comes out of DNA in the form of mRNA. mRNA is the instructions for how to build a molecule, usually a protein. And then that protein gets packaged so it's usually built in the rough ER. Here we've labeled the inside of the ER with blue and the outside of the ER with kind of this tan color, right? So it's phospholipid bilayer, remember? So that protein is constructed. It's going to move from the rough ER in a vesicle, that membrane bubble, to the Golgi, where the Golgi will make any final edits, maybe add on some carbohydrates, change it around, anything that it needs to do. And then once it's finally edited, the Golgi will send out another vesicle with the final protein, and that vesicle fuses with the plasma membrane. And then whatever proteins were embedded in that vesicle end up in the plasma membrane, right? And if we want some proteins just on the outside of the cell, we embed them on the inside of the vesicle, because as the vesicle fuses, the inside of the vesicle becomes the outside of the cell membrane. Okay, so that's how we get proteins on the cell surface. And we can add proteins by adding vesicles, and we can take away proteins if we don't want certain proteins here anymore. We don't need these receptors. We can take a vesicle in and then degrade those proteins, right? So it goes both ways. So we can add proteins by putting vesicle on, and take proteins away by removing vesicles. So that's our quick overview of membrane structure. We'll talk later about how the membrane works, and specializations of the membrane, and how stuff moves in and out of that membrane as well. But the key here is that there are lots of proteins embedded in that membrane. They have a wide variety of functions, and we just covered the main categories of proteins that are embedded in the membrane.